Okay, let's hope. Okay, any other questions? Okay, so then you can construct something like this. So this would be the multiple sequence alignment, and again here you see how this was built up. So first these were aligned, then these were aligned, then you combine these two sets, and then last you align all of them. Yeah? And so that's called progressive alignment. Now there's more complicated and also more precise ways to do this. This is one of the most simple ways. Still very much used. So this is cluster W. Has this multiple sequence alignment strategy, and in our group, so Beringa has also developed several multiple sequence alignment tools with slightly different methods. So, but now we have this multiple sequence alignment tool. Do we want to put more weight on motifs? So I'll just introduce what motifs are. So for example here, we have a typical motif. So if we look here at the protein structure, we see here in yellow-orangey, we see part of the sequence, or part of the protein chain here, that is colored yellow, and that is has a specific motif. And the ones that are specific are in dark yellow. And you see them also here in dark yellow in this multiple sequence alignment motif. Now what does this do? This is actually a copper binding motif. And these residues are almost there, at this kind of same exact spacing from each other. And typically in this 3D structure would also have the same orientation to this copper here. So especially this pocket here. These residues really need to be at a certain distance from each other to be able to bind this copper. If you see this back, so if you here you have a whole sequence family, and here you already see a multiple sequence alignment, and you see that indeed the positions and the motifs mostly really align with each other. This is still, by the way, done by a method, and so if you really wanted to align this better, you could actually put this, so you see this protein here, but you may actually want to put a gap between the P and the H here to get this motif aligned even better. So it's actually not completely optimal. Now this is actually a difficult problem. So to, and this of course that you want these residues on top of each other is important, because typically you want to know if the function of copper binding is still there in your protein. So you want to see if that aligns with the other sequences. But this is not always possible. So this is another case, and here we have a HIV protein, and this has glycosylation sites. Now, glycosylation motif is mentioned here. So it's an N on the first position. Then on the second position, the only restraint is not a proline. That's what these curly brackets here mean. And then on the third position, you get either a serine or a threonine, so SOT. And on the fourth position, again, it's not a protein. So it can be any amino acid, but not a protein. So here you can see that these motifs here, with typically an N on the first position, and then a T or an S on the third position, are annotated. And this is a crystal omega, so that's the, I mentioned earlier, crystal W. Crystal omega is the kind of next generation of crystal W. And you see that these motifs are very poorly aligned. They don't form nicely on top of each other in the homolog. So this is not something, even though you think like, okay, there's N, so these methods should try and align these on top of each other, that doesn't in practice happen. Also because there's other residues there. So now you can't really see if these motifs are nicely conserved in the sequence, which is what you want to know. This is a different region of the same protein. You see here actually it does a much better job. And so you see here actually that the residues in this case, or the motifs, are very nicely aligned. So the question is can we improve upon this? And now this is where Maurits Dijkstra came in, and he generated a protocol which you can really do better here. And I'll show you that in a minute. So imagine that you have three sequences here, yeah? So we have sequence 1, sequence 2, and sequence 3. And we have annotations, so we have amino acid sequence, yeah, that's what you use to align. This is what you put in your dynamic programming matrix. But now we have additional annotations. Now we have here a secondary structure. So this is what I said earlier. You may actually want to try and align part of helices, or E is beta strand and C is coil or loop. And you may actually want to align residues with the same annotation on top of each other. What we're now really interested in is the motifs. So here also there's a track, an annotation track for each sequence that indicates whether there's glycosylation motif in that sequence. So if you look back, you indeed see here that there's an N on the first position and a T or an S on the second position. Every time you have these M's here in this track. You see that? Yeah, so these M's here just indicate whether there's this glycosylation motif or not at a specific point in the sequence. And so now what you can do is you can actually put additional weight on your positions where you have motifs, if the motifs are aligned properly. So you can go back to your dynamic programming matrix, and then if you look up the annotations, and you have a specific score, so if you fill in one of your cells, you now actually can put extra weight or extra scores if two motif positions align also with the right position. So if the first motif position aligns in another sequence to also the first motif position. Yeah? And then you get extra weight, and this basically forces these motifs to align more nicely together. And so if you do this, you see now an alignment where indeed the motifs are nicely aligned on top of each other in the multiple sequence alignment. Yeah, so the trick of the weight is actually done in a pairwise step, so where you fill in this matrix and you add weight. So that's just what you're used to in the dynamic programming. And then progressively you go through this, as I showed before, that you have first the pairwise alignments and then you add additional sequences in a kind of progressive way to this multiple sequence alignment. And here is the motif explained again, but I went through it already earlier. So this is the figure you actually need to know for which you need to revise for the exam. Yeah? So this is part. And so this is from a different paper, and it really focuses on the motif aware. And what you see here is this kind of gradual improvement of this algorithm. So you see that if we put this algorithm on stronger, we put this motif weight on stronger, we actually get better alignments. So here's normal praline, and it does a pretty poor job. You see not many of the motifs are aligned. But if you then go to MA praline, so this is the motif aware praline, you see that the motifs start to really be aligned much, much better. This is just by putting a bit of weight on this. And then you get it even better if you only zoom in, so you kind of decouple this V1 difficult to align region and you just try to align this. You get even a better result. But you see that only doing that is not enough. Then again, so if you have the old standard praline without this motif weight, you don't actually get an improvement at all. So this can also be done for other proteins. Multiple motifs can be used, and this is now just in colors, but you see that the other methods still have a lot of trouble aligning it. And you see here that the Witty motif score, you really get a much better alignment. And you can also do this for DNA. So if you have a DNA, they have motifs as well, because proteins actually bind to the DNA, especially in the promoter region. You may actually get transcription factor binding sites. And they have also very specific motifs, but then DNA motifs, yeah, so just with 4 letters. And that's what you see here. And the motifs here are annotated in different colors. So for example, here's a motif, a transcription factor binding motif in green here, you see GATA here. So you see here different transcription factor binding motifs, also in red here. And you see that combined, which is also a version of this motif-aware alignment method, but now for DNA sequences, can really make a good alignment and really align two motifs on top of each other. That's already this part of the highlights. For this paper, so we said for the highlights you revise the abstract. For the exam you don't necessarily need to read the whole paper. This is the abstract, so it's tiny. The paper is 1 page, so I suggest here that you do read the paper, otherwise you don't even have enough information. And that will also help you to understand this figure and the caption, of course. That doesn't mean we will show you this figure necessarily on the exam. We may give you something new, but just so you know. Are there any questions on this? No? Okay, then we'll have, I think, how long a break shall we do? How long do you need? 10 minutes, so we'll be here back at 5 to 5:30? Yeah, 5:30 we'll start.
Oh, no. Oh.